Assalamu alaikum wa rahmatullahi wa barakatuh. Our seminar today is about atypical viral pneumonia created by Dayvid Bahnan, Hisham Saadallah, Mohammed Jaseem, supervised by Dr. Karam Agha. Let's start. The most common causes of community-acquired viral pneumonias are influenza types A and B, the respiratory syncytial viruses, human metapneumovirus, adenovirus, rhinoviruses, rubella virus, and varicella virus. Nearly all of these agents also cause upper respiratory tract infection, common cold. Pathogenic mechanism, how the disease starts. These pathologic viruses share a propensity to infect and damage respiratory epithelium, producing an inflammatory response. When the process extends to alveoli, there is usually interstitial inflammation, but some outpouring of fluid into alveolar spaces may also occur, so that on chest films the changes may mimic those of bacterial pneumonia. As a result, it is not possible to distinguish bacterial and viral pneumonia based on radiologic appearance alone. Moreover, damage leading to necrosis of the respiratory epithelium inhibits mucociliary clearance and predisposes to secondary bacterial infection. Such serious complications of viral infections are more likely in infants, old adults, malnourished patients, alcoholic, and immunosuppressed individuals. What are morphology? The morphologic patterns in viral pneumonias are similar. The process may be patchy or it may involve whole loops, bilaterally or unilaterally. Macroscopically, the affected areas are red blue, congested, and subcrepitant, meaning crackle sound. On histologic examination, the inflammatory reaction is largely confined to the walls of alveoli. The septa are widened and edematous. They usually contain a mononuclear inflammatory infiltrate of lymphocytes, macrophages, and occasionally plasma cells. In the classic case, alveolar spaces in viral pneumonias are free of cellular exudate. In severe cases, however, full-blown diffuse alveolar damage in hairy membranes may develop. In less severe, uncomplicated cases, subsidence of the disease is followed by reconstitution, meaning rebuilding of normal architecture. Superimposed bacterial infection, as expected, result in a mixed histologic picture. Picture description: viral pneumonia. The thickened alveolar wall are infiltrated with lymphocyte and some plasma cells, which are spilling over into alveolar spaces. Note the focal alveolar edema, center, and early fibrosis upper right. Clinical features: the clinical course of viral pneumonia is extremely varied. It may masquerade mean تتنكر as a severe upper respiratory tract infection or chest cold that goes undiagnosed or manifests as a fulminant life-threatening infection in immunocompromised patients. The initial presentation usually is that of an acute non-specific febrile illness characterized by fever, headache, and malaise, and later cough with minimal sputum. The localization of the inflammatory exudate to the alveolar walls prevents oxygenation of blood flow through the affected air spaces, which in turn causes mismatch of ventilation and perfusion. As a result, the degree of respiratory distress often seems out of proportion to the physical and radiographic findings. Identifying the causative agent can be difficult. influenza infection. Perhaps no other com- communicable disorder causes as much public concern in the developed world as the threat of an influenza epidemic. The influenza virus is a single-strand RNA virus bound by nucleoprotein that determines the virus type A, B, or C. The viral surface has lipid bilayer containing the viral hemagglutinin and neuraminidase, which determine the subtype, for example, H1N1, H3N2. Host antibodies to the hemagglutinin and neuraminidase prevent and ameliorate respectively future infection with the influenza virus. The type A viruses infect humans, pigs, horses, and birds, and are the major cause of pandemic and epidemic influenza infection. The virus is cytopathic to respiratory epithelium, leaving a bare-based membrane on which Staphylococcus and other organisms grow. The disease is seen every year and in the temperate climate mean مناخ معتدل, usually during winter months. Viral spread is by droplets and spread is increased by close contact or crowding. The disease is only usually fatal in its acute stage if there is underlying cardiopulmonary disease. Death is usually due to staphylococcal pneumonia. Epidemics of influenza occur through mutation of the hemagglutinin and neuraminidase antigen that allow the virus to escape most host antibodies, antigenic drift. Pandemics, which last longer and are more widespread than epidemic, may occur when both the hemagglutinin and neuraminidase are replaced through recombination of RNA segments with those of animal viruses, making all animals susceptible to the new influenza virus. Antigenic shift. Commercially available influenza vaccines provide reasonable protection against the disease, especially in vulnerable infants and in older adults. Insight into future pandemics has come from studying those past. DNA analysis of viral genomes retrieved from the lungs of soldiers who died in the Great 1918 influenza pandemic that killed 20 million to 40 million individuals worldwide identified swan influenza sequences consistent with this virus having its origin in an antigenic shift. The first flu pandemic of the century in 2009 also resulted from an antigenic shift involving a virus of swan origin. It caused particularly severe infection in young adults, apparently because older adults had antibodies against past influenza strains that conveyed partial protection. Comorbidities such as diabetes, heart disease, lung disease, and immunosuppression also were associated with a higher risk for severe infection. What then might be the source of the next great pandemic? One concern is centered on avian influenza, which normally affects birds, and mean influenza الطيور. On avian strain type H5N1 has spread throughout the world in wild and domestic birds. As of September 2015, approximately 700 H5N1 influenza virus infections in and 14 adults in humans from 15 countries have been reported to the World Health Organization. Nearly all cases has been acquired by close contact with domestic birds. Most deaths resulted from pneumonia. Fortunately, the transmission of the current H5N1 avian virus is inefficient. However, if H5N1 influenza recombines with an influenza that is highly infectious for humans, a strain might emerge that's capable of sustained human-to-human transmission and thus of causing the next great pandemic. Oh, we have a question. My colleague Hisham will continue. Let's listen.
Thank you, my colleague David. Assalamu alaikum. Firstly, welcome everyone hearing our seminar. My name is Hisham Saadallah. Now I will talk about an important subject and every medical student must to have headline about it. So, the answer for Robin's question is COVID-19. The number of infections in the world reach 4 million according to the World Health Organization, and the number of deaths is approximately 300,000 until the date of writing this seminar in May 2020. Coronavirus. So, coronavirus first emerged in the mid-1960s, and there are 7 different versions of the virus broken into 4 groups: alpha, beta, gamma, and delta. Now, what is common in human coronaviruses? There are many coronaviruses that attack the human body, like 229E alpha coronavirus, also NL63 alpha coronavirus, also there is OC43 beta coronavirus, and HKU1 beta coronavirus. Other human coronaviruses. First one, MERS coronavirus, the beta coronavirus that causes Middle East respiratory syndrome. So every letter means word. M means middle, E, East, R, respiratory, and S means syndrome. Second one, SARS coronavirus, the beta coronavirus that causes severe acute respiratory syndrome, or SARS. Third one, SARS coronavirus 2, the novel coronavirus that causes coronavirus disease 2019 or COVID-19. This figure illustrates the types of coronavirus, where the alpha coronavirus many types, but the common types are 229E and NL63. Also, the beta coronavirus many types, but the common types are OC43 and HKU1. And there are other human coronavirus like MERS coronavirus or Middle East respiratory syndrome, and SARS coronavirus, or called severe acute respiratory syndrome. This diagram illustrates the types of coronavirus and the causes that transmit the virus to human. So I will start with the SARS, where it is transmitted to human through type of wild cat called civet cat. In the next slide I will show you a cat photo. Now the second type is MERS or Middle East respiratory syndrome, where it is transmitted to human through meat and camel milk. And last type and modern type, which is novel coronavirus or COVID-19. The causative is not yet known, but it is believed that snakes and bats have a role in its transmission to humans. The question that arises: why do scientists and doctors suspect snakes and bats in particularly and not other animals? And the answer is there is a glycoprotein on the surface of the virus. This glycoprotein helps the virus to adhere to human cell. So scientists have found this glycoprotein is similar to the glycoprotein of virus that infect the bats. So they thought bats were the reason to transmit coronavirus to humans. They also found the RNA of virus is similar to the RNA for the virus that infect snakes. So scientists and doctors believed that snakes were the other cause of transmitting the virus to humans. This picture shows MERS coronavirus, where the virus is transmitted to human through meat and camel milk, as shown in the picture. This wildcat called Dayvid is responsible for transmitting the SARS virus to human. Is the coronavirus disease the same as SARS? The answer is no. The virus that causes COVID-19 and the one that causes the outbreak of severe acute respiratory syndrome SARS in 2003 and related to each other genetically, but the diseases they cause are quite different. So COVID-19 spreads faster, but it is not as severe as SARS. الشعوب العربية ومن ضمنهم العراق يستغربون من بلد الصين وأكلهم للفئران والأفاعي والقوارض التي تشمئز لها النفوس. لكن لو نرجع بالتاريخ راح نعرف أن السبب من أكلهم هذه الأكلات هو الصين سابقا في خمسينيات القرن الماضي عانت من مجاعة كارثية، مما جعلهم يأكلون هذه الحيوانات، وهذا جعلهم يتعودون على أكلها إلى يومنا هذا، وصار جزء لا يتجزأ على موائدهم. وإذا تلاحظون هذه الصورة وكيفية البؤس والفقر اللي كانوا يعيشون أطفال الصين وعوائلهم، لذلك قالوا تحشيش عليهم لأن حياتنا كلها made in China اللي نقلد عليهم. Now SARS, Severe Acute Respiratory Syndrome, SARS, is an infectious condition caused by a coronavirus. It emerged from Guangdong province in China in November 2002 and resulted in widely publicized outbreaks in Hong Kong, Vietnam, and Singapore. Now, common symptoms of SARS. First one, fever. Second one, cough or dry cough. Third one, SOB or shortness of breath. This will lead to pneumonia or ARDS, acute respiratory distress syndrome. So what is the difference between pneumonia and ARDS in general? The answer is pneumonia affects a specific region of the lung, while ARDS affects the entire lungs. And lastly, the pathogenesis. A human infection with SARS coronavirus similarly led to an acute inflammatory response. In human, SARS coronavirus caused diffuse alveolar damage and deposition of fibrin with macrophage and giant cell infiltration in lung tissue. Acute hepatitis have been widely reported and may be a result of immune-mediated rather than direct SARS coronavirus viral damage. The main picture that seems to emerge from the majority of the studies is that interleukin 6 and interleukin 8 arise during SARS coronavirus infection. Interleukin 6 is secreted by macrophages and T lymphocytes and causes fever-induced acute phase proteins and activates lymphocytes. Interleukin 8 is secreted by monocytes, macrophages, fibroblasts, and keratinocytes, and is a chemoattractant and enhances access for neutrophil to the site of infection. By the pathological features were dominated diffuse alveolar damage with the presence of multinucleated pneumocysts, fibrogranulation tissue, proliferation in small airways and air spaces, bronchiolitis, obliterans, organizing pneumonia-like lesions in subpleural locations was also seen in some patients. And this reference of our seminar. Thank you for listening.